Hello, welcome back. So we're going to talk quickly about, I'm going to actually go back over here just to remind you. We're going to talk about these different levels of control in eukaryotic gene expression. So we're going to start with pre-transcriptional control. And I love this picture because it really shows pre-transcriptional control. So the way DNA is packaged inside of the cells, inside of the nucleus, actually keeps genes turned off. DNA can also be methylated, which will keep the genes turned off. And genes located within heterochromatin are also inactive. And so this includes bar bodies, which are found in mammalian females. These inactivate the X chromosome in females. So all females in every single cell have two X chromosomes. And I should say in general, because there's always those anomalies, but let's just in general. Within the cell itself, one of those X chromosomes will be randomly turned off or inactivated. And you can see this best in calico cat coloration. So the color, the allele for the fur color in calico cats is found on this X chromosome. And so some calico cats have this allele for orange fur color. And if that is turned on, then the cat is going to have that orange coloration. If they also have an allele for black color, in those cells, they're going to have black coloration. And so you end up with this black and orange coat on this calico cat. And so this is pre-transcriptional control. Another mechanism of pre-transcriptional control is the way those genes are packed. So if they are packed in chromatin, then they're not going to be able to be transcribed. So until they unzip, they can't get transcribed. And so that's another mechanism of pre-transcriptional control. So then let's look at transcriptional control. I don't have a beautiful picture for this. This is a picture from your textbook. But transcriptional control then depends on interactions between proteins and the DNA sequence. So proteins called transcription factors and activators are going to determine if the RNA polymerase can actually bind to the promoter region and transcribe. So for transcriptional control, we're going to need activators and transcription factors. That are going to influence the ability for the protein or the messenger RNA to be produced. So transcriptional activators are proteins that increase transcription. And if those activators are not available, then transcription is not going to be done effectively. So then we have post-transcriptional control. So I'm going to put post-transcriptional control would be the addition of that poly A tail, the addition of the guanine cap, the removal of the introns, all of the mechanisms that produce that mature messenger RNA are all part of that post-transcriptional control. If you don't do that, then you're not going to be able to send that messenger RNA out into the cytoplasm where it can be translated on the ribosome. We have translational control. So translational control is going to be dependent on the length of the guanine cap and the poly A tail to determine how long that messenger RNA can be translated before it is broken down. So very short guanine cap and poly A tails are going to only be able to be translated 1 or 2 times, versus if they're very long, then they can be translated multiple times. And then we have post-translational control. So post-translational control is going to occur after you've produced the protein. The protein then is going to be inactive until we activate it. And so we can activate proteins by cleaving a portion of the protein off. An example, in our stomach, we have a protein that digests, we have a protein enzyme that digests proteins. So when we eat meat, the proteins start digesting in the stomach, okay? Start getting chemically digested in the stomach. So the protein that does this is called pepsinogen. But pepsinogen, in a nutshell, is inactive. It has to come in contact with hydrochloric acid. Oops, sorry, that was not a C. So hydrochloric acid cleaves a portion of that pepsinogen, producing the active form known as pepsin. And so that is post-translational control. So the last thing we're going to talk about is gene mutations. And so I'm going to quickly go over what a mutation is. A mutation is a spontaneous change in the DNA. So, well, is a change in the DNA. I shouldn't say it's spontaneous, but it can be a spontaneous change in the DNA. It also can occur due to environmental influences. So DNA replication is not error-free. There are mutations that occur, but the mutations are very rare. And this is because we have DNA polymerase, which reads and proofreads the new DNA that we produce. And so if there's a mistake, then we have enzymes that cut out those mistake regions and put in the proper nucleotides. So there are very few mutations in replication that are spontaneous, but they can occur. Spontaneous mutations do occur. And the more common way that mutations occur is through mutagens. Mutagens are environmental influences, such as chemicals, that can influence DNA or can cause changes in DNA. So radiation, X-rays, UV rays can cause different problems, different mutations within DNA. Chemicals, so I say chemicals. Cigarette smoke is an example of a chemical. Pesticides, those are chemicals that can cause mutations in our DNA. And then we have DNA repair enzymes that are constantly checking to make sure our DNA is correct. Remember in chapter 5, we talked about the different mechanisms, the different control checkpoints that we have. CDK and cyclins are constantly checking to make sure everything looks right before we go on in the cell cycle. So we don't have mutations, but mutations do occur. And when they occur, they can cause different problems. So what are the types of mutations, or what are other causes of mutations? Transposons. Transposons are not chemicals, so they're not induced. They're also not spontaneous. Transposons are jumping genes. Jumping genes are genes that can move from one region of DNA to another region of DNA, or can move from one DNA sequence to a completely different chromosome. And so this can alter gene expression, and this can cause all kinds of problems within the genes of the organism, because it can either turn on or turn off different genes. And so we can see this. Hold on, let me see. Do I have a beautiful picture? I do. So let's go to this one. We can see these jumping genes, or these transposons, when we look at corn. So corn typically has this purple pigment here. So this is a specific type of corn, right? But when you have a transposon within that purple pigment, all of a sudden you get these yellow and these like reddish corn kernels, okay? So you don't get that purple pigment anymore. Now you get this unique pigment coloration because of that transposon. So that's an example of what a transposon does. So point mutations. Point mutations can be caused either due to a spontaneous mutation or due to an induced mutation. And a point mutation is when one single nucleotide changes. So this can cause different effects depending on which nucleotide was changed. So it could not affect the organism or the protein at all. It could produce a protein that is incomplete, or it could produce a protein that has a new shape, because now we have a totally different amino acid being produced. So here's an example of a base substitution where we end up with a missense mutation. So I didn't write this down, so let me just go to the slide before. So this here, base substitution, is typically called a nonsense mutation. If it changes the amino acid, it's called a missense mutation. And then if it has no effect, it's called a silent mutation. So sickle cell disease is actually caused by a missense mutation. So here is what the DNA typically looks like, and then this is the messenger RNA. Now, if you look over here, there's one nucleotide difference in the entire sequence. So this is not the whole sequence, of course, of the hemoglobin. But if you look right here, there's an A, and normally you have a T. So this T here has been changed. It has converted to an A. And because of that one change, when you produce the messenger RNA, instead of having GAA, which codes for glutamate, and now you have GUA, and that codes for valine. And that one amino acid difference causes the blood cell, the hemoglobin, to change shape, which causes the entire cell to change shape. And then you have sickle cell disease or sickle cell trait, depending on if you have two alleles or not. Frameshift mutations then differ in that you are either inserting or deleting DNA. And again, this could be induced or spontaneous. The result then occurs, or the result that occurs is either a completely non-functional protein or a very differently functional protein. So I always use the sentence, the cat ate the rat. It's the easiest way to explain it. We read our DNA in 3 nucleotides, right? So the cat ate the rat is a sentence that we all can understand. So let's just take out the C. We're taking the C away. And now when we read it, it says the atta tet her at. That is a non-functional sentence. It doesn't make any sense. And the only thing we did was we deleted 1 nucleotide. So we changed the frame in which we were reading. So we still read 3 nucleotides at a time, or 3 letters at a time, but those 3 letters are all different. So these amino acids here, this section that codes for an amino acid and this amino acid are going to be different than the amino acids up here. And this doesn't even code for an amino acid because there's only 2 nucleotides. So here are the different types, again, of point mutations. This is what our normal DNA looks like. This is a base substitution. There are 3 types of base substitutions. You either have a nonsense, missense, or I'm sorry, that should be an S, huh? Or a silent. Then we have addition, where we are adding a nucleotide in, or deletion, where we're taking a nucleotide out. So in this case, we have methionine, phenylalanine, threonine. Here we have methionine, leucine, threonine, arginine, isoleucine. So this would be a missense here. And then down here, we have taken 1 nucleotide out. Or no, we've added 1 nucleotide in. So we have methionine, phenylalanine, but now we have aspargine, leucine, and aspargine again. So we've changed the amino acid sequence. So the structure has changed, so it's going to be differently functional. And down here, we have deleted 1 nucleotide. So now we have methionine, leucine, arginine, and glutamine. So again, we've changed everything down from that 1 mutation. And so these are going to end up being either non-functional or very differently functional than the original protein that was there. And that is the end of our video. So I'm going to upload this, and I will talk to you a little later. Bye.